the warm shallows of an ancient sea, sunlight bends through water that is neither ocean nor lake, but something in between a tidal margin, not yet defined by continents or coasts. Here, beneath a ceiling of flickering light and silt, clusters of green algae drift softly tethered, not by roots, but by the rhythms of current and wave. Theirs is a world held in suspension, not quite sea, not quite land, where buoyancy protects them from gravity and salt does not yet sting the skin. Their filaments drift with an ease that betrays no urgency, photosynthesizing impulses as the sun moves overhead. Each cell filled with chlorophyll and the silent memory of cyanobacterial ancestors converts light into sugar, quietly rewriting carbon into form. Among them float the charophytes' algae of strange promise. Their bodies are simple, often filamentous or disc-shaped, but within their tissues lie traits that do not belong entirely to the sea. Their cell walls are reinforced with cellulose, their division planes controlled by microtubules that form in patterns curiously similar to those found in land plants millions of years hence. They live in liminal pools, sometimes left stranded for hours as tides recede and puddles shrink. In these fleeting moments, desiccation begins the subtle cracking of membranes, the whisper of air against surfaces evolved for water. And yet they endure. Some form protective sheaths, others dry down and wait. The difference between survival and dissolution lies in timing, in the quiet choreography of wet and dry, of swelling and collapse. These shorelines are not yet green in the way we understand it. There is no forest, no grass, not even moss. Only microbial mats and thin films of algae stain the edges of rock and sediment, shifting color slightly under the slow abrasion of wave. Stone dominates this world raw, unsoiled, and bare, unmarked by roots or rain-fed erosion. The continents are lifeless monuments rising above the sea, weathered only by wind and time. Yet in these few zones where water lingers on rocks that stay damp in rivulets warmed by afternoon light, the algae begin to press closer, not in conquest, but in passive accumulation. Spores drift and settle, filaments stretch. What clings survives. What floats away is forgotten. No one moment defines the transition. There is no single cell that becomes a plant. Instead, it is a gradient, a shifting blur of generations, accumulating microscopic advantages. One lineage produces a slightly thicker wall. Another survives drying an hour longer. Some divide asymmetrically. Others form protective layers that hint at the earliest cuticles. These traits are not yet coordinated. There is no plan, no architecture. Only an ongoing experiment in surviving absence, the absence of water, of salt, of buoyant suspension. In losing the sea life begins to remember land. The irony is slow and quiet, that the very medium which first gave rise to life, the protective soup of ancient oceans, would one day become insufficient. That evolution in its inexorable drift would favor those who endured exposure, who were dried and not destroyed. That survival would hinge not on mobility or speed, but on the ability to remain still and persist through thirst. A kind of patience takes root long before roots exist, and still the algae do not move with purpose. They are carried. Their cells do not stretch toward the land so much as they endure it when it arrives through splash, through wind-borne droplets, through tides pushed further inland by moon and tectonic rearrangement. In some sheltered pools, salinity decreases over centuries. Rainfall gathers more frequently. The rock begins to change, no longer a dead host, but a slowly dissolving surface. Microscopic pits and hollows offer shelter. Minerals begin to leach, providing the faintest hint of nutrition beyond the sea. There is a poetic irony in this too, that stone, the most inert of substances, becomes the medium through which life first contemplates permanence. In water, organisms drift and disperse. On stone, they must cling. Attachment becomes an evolutionary act. The edge of a rock is no longer just a resting place, it is a frontier, a challenge. To remain here is to resist being washed away. It is to endure light without shade, dryness without pause, gravity without lift. And so algae begin to favor the margins. The zones where tides reach but do not remain, where mist falls but does not pool, where every advantage is fleeting and every moment of survival is a rehearsal for independence. From the vantage point of deep time, these moments blur. 10,000 years become a sigh, 1,000,000 a breath. Yet in these long silences, adaptation accumulates. Each trait, however slight, carries forward a lineage, a memory of conditions barely endured. And in this way, the green pigment of algae forged in water prepares to lift its face toward the sky, not yet as plant, not yet as anything new, but as possibility, as rehearsal. There is mystery here too, quiet and unresolved. Why did only certain algal groups begin this slow procession toward land, while others equally complex, equally capable, remained forever aquatic? Was it the shape of the cell wall, the timing of division, the composition of mucilage, or was it accident? 1,000 tiny coincidences strung across 100 million years, science draws outlines around these questions, but rarely fills them. Even now, the fossil record for early landward algae is fragmentary. Soft-bodied and ephemeral, they leave behind little more than patterns in stone and the biochemical echoes of ancient lipids. And yet they were here. They must have been. A final observation whispered through time for all the complexity of later plants, the towering ferns, the branching trees, the flowering marvels of later eons. It is these algae who first imagine what it means to leave the water, who first learn that light can be encountered without submersion, who discover without knowing that land can hold life if it is approached slowly enough and long enough and with quiet resilience. They are not in a hurry. Nothing in this story is. The shore remains silent, sculpted by wind and tide, indifferent to the ambitions of living matter. Yet, at its edge, where the shimmer of water retreats and returns in uneven rhythms, certain algae begin to persist with new tenacity. These are not the free-floating colonies of the open shallows, but something quieter clinging already shaped by an intimacy with rock. Among them are the charophytes, green algae of a lineage closer to moss than kelp, forming mats that adhere to stone with subtle threads. Not roots, but precursors, not organs, but improvisations. Their forms are simple, but their biology speaks a more elaborate language. Plasmodesmata, linking cells. Phragmoplasts, guiding cell division, and the first murmurs of genes that will one day script stems, pores, and spores. They have no leaves, no bark, no flowers yet. Within them lives the architecture of forests, not yet imagined. These algae remember rock, not as surface, but as shelter challenge and partner. They do not merely rest on it. They form biochemical conversations with its surface, releasing enzymes to tease apart, silicates absorbing trace metals and adjusting to its dryness with layers of mucilage that shimmer faintly when exposed to sun. In the laboratory, modern charophytes are watched under microscopes, their chloroplasts spinning gently inside translucent walls. But here in the blurred swamps of the Silurian and late Ordovician, they simply endure, exposed to wind that carries saltless dust and sun that burns without mercy. They do not scatter when tides recede. They tighten, curl inward, shield their cores with sugar polymers and cell wall reinforcements. These are not passive organisms. Their stillness is an action. Their cling is intent. In their genome, a strange resilience accumulates: desiccation, tolerance, antioxidant enzymes, UV-absorbing pigments. Not all survive, but those that do carry forward, not a strategy, but a memory, not a thought, but an encoded patience. The irony, slow and unnoticed, lies in how evolution rewards those who do not move, those who stay behind, while others drift away. To remain affixed is to invite dryness, to risk sunlight unfiltered by water, and yet in doing so, these algae begin to test conditions that will define all future terrestrial life: evaporation, exposure, irradiation. They do not know it, but their survival sketches, the contours of later leaves, later roots, later breaths of land-bound air. Their simplicity masks their importance. No fossils preserve their softness in perfect form. What remains is chemical fragments of alkanes' isotopic signatures in ancient mudstones, molecular hints of cutin-like compounds long before vascular tissues. The rock becomes the record not through imprint, but through reaction, and so we learn of them not from their presence, but from what they leave behind when they die. A trace of lipids, a roughened layer of sediment, a change in the ratio of carbon isotopes, slight persistent telling. There is a gentle mystery in how such algae adapted to life on hard surfaces before land existed as a true habitat. What drove their shift from water column to rock? Was it competition? Was it nutrient access, light exposure, or the accidental currents of shallow pools evaporating faster than usual? Or perhaps it was simply that the rock was there, stable, unmoving, a surface to hold against the indifferent tides. Scientists reconstruct their likely habitats from thin layers of shale, from fossilized shorelines, now lifted into mountains from modern analogues in desert crusts, where algae still cling to stones and survive dehydration cycles daily. And yet certainty is elusive. No complete lineage can be drawn from charophyte to moss, only patterns, only likelihood. Evolution does not leave blueprints, only shadows of construction. Meanwhile, as the algae hold fast, the rock beneath them is changing too. Lichen-like associations begin to appear, delicate alliances between algae and fungi that soften the surface, pulling minerals from the matrix, creating crevices that hold water a little longer. These biofilms are primitive, and their partnerships remain uncertain in origin, but they signal a profound shift. The beginning of mutualism, the idea that life can extend itself by joining with other life. Mycorrhizal threads will one day enter roots, but here in this earliest version, the principle is already seeded. Fungi may offer shelter from ultraviolet rays. Algae offer photosynthetes. The exchange is not efficient, not refined, but it is enough to persist. Enough to endure one more dry day, one more cycle of ebb and sun. There is no rush. The green edge of the world expands not with conquest, but with repetition. One generation clings slightly better. One colony stretches further up the stone. One mutation causes a thicker mucilage sheath. Another perhaps enables delayed cell division during dry phases. It is a choreography played out in the margins, never noticed at the time. Only in retrospect does it reveal direction. The irony is soft but firm that what will become a tree, a forest, a planetary transformation begins not with branches, but with the ability to dry and wait, with the discipline of stillness. Even now in highland seeps and alpine tundras, modern relatives of these algae remain whispering perhaps of what they once risked. Their chloroplasts still rotate. Their filaments still adhere. Their genes still speak the language of thresholds, wet and dry, dark and light attachment and release. Evolution at this stage does not seek dominance. It seeks compatibility between membrane and mineral, between cell and light, between water and its absence. And always quietly, the question lingers, why here? Why these particular algae? The sea is full of photosynthetic life, red algae, brown algae, cyanobacteria, all prolific, all ancient. But only this lineage, these green charophytes, begin to edge toward the possibility of land. Not because they are superior, not because they choose, but because in the slow arithmetic of mutation and time, their traits happen to align with the conditions of terrestrial life. This is not destiny, it is filtration. The sea sifts possibility through the sieve of stone, and these algae remain. Thus, they become the hinge, between fluid and fixed, between the nourishment of water and the exposure of air, between the familiarity of submersion and the risk of light untempered. 1,000 million years after life begins, algae come to remember stone not as a refuge, but as an invitation. And in remembering, they begin to imagine, what it might mean to stay. Above the green films clinging to stone, the sun bears down with unfiltered light, radiant, merciless, and constant. In the open ocean, light arrives in softened bands, scattered and refracted, buffered by depth and motion. But here on the edge of air, the full force of solar radiation strikes without resistance. Ultraviolet rays pierce membranes. Photons excite and destabilize. The protective shroud of water, once a gentle filter, now lies behind, and what was once nourishing light becomes both sustenance and threat. These algae, green and humble, have not evolved for such intensity, and yet over countless generations they begin to respond. Pigments deepen, flavonoids accumulate. Molecular shields form not visible armor, but cellular strategies coded into enzymes and proteins. Mutations once neutral or irrelevant now become decisive. Those that survive do not do so by thriving, but by enduring. Theirs is not adaptation through conquest, but through the slow accrual of tolerance. This is the crucible of early terrestrial existence: sunlight without shelter, desiccation without reprieve, gravity without buoyancy. The removal of water does more than dry the surface. It reveals the physical laws that water once concealed. Gravity, long irrelevant, now begins to assert itself. A floating filament in the shallows weighs nothing. Laid bare on stone, it slumps, collapses under its own softness. Cells deform. Structures sag. Shapes become contingent on strength. The very architecture of life begins to shift. To remain upright, even at the scale of millimeters, demands a new kind of internal scaffolding. Microtubules align, cell walls thicken, cellulose is laid down in crisscross spirals. The earliest clues of terrestrial form emerge not from ambition, but from necessity. To face the sun, even slightly, requires resistance to collapse. The irony is quiet, but clear, that the urge to reach upward is born first from the need not to fall. And yet the transition is not linear. There are reversals. Entire lineages that explore the margins return to water, their tentative adaptations discarded. There are fossils of plants that once bore features suited to land, cuticle-like coatings, desiccation-resistant spores, only to vanish from terrestrial strata, reappearing later in aquatic forms. Evolution at this stage is not progress, but oscillation. The boundary between water and land is porous, not only physically but genetically. Horizontal gene transfers occur. Fungal genes seep into algal genomes. The vocabulary of stress response is borrowed, refined, and reshuffled across domains of life. These are not conscious transactions, but molecular exchanges shaped by time and exposure. The idea of what constitutes a plant, what defines its separateness from fungus, bacterium, or algae becomes less a matter of taxonomy and more a matter of context. Scientific evidence for these early transitions remains partial. Fossilized microspores, trilet or otherwise ornamented, are among the few durable traces, their patterns suggesting airborne dispersal, their chemistry hinting at protective coatings, now called sporopollenin. This substance, among the most chemically inert organic compounds known, offers clues to how early cells withstood both drying and ultraviolet light. It is in essence a molecular answer to sun and wind. The earliest land plants did not evolve new forms overnight. They evolved ways to survive the absence of water. These ways were chemical first, structural second. The ability to photosynthesize was never in question. The challenge was to do so in air with light unmediated and water no longer surrounding every surface. In laboratories, researchers simulate these conditions, charophytes, subjected to cycles of dehydration and rehydration. Their gene expression monitored, their cell walls observed under electron microscopes. They find that many of the mechanisms once assumed to be exclusive to land plants already exist in rudimentary, scattered, but unmistakable forms in their aquatic relatives. The division is not absolute, it is temporal. The ancestors of mosses and liverworts were not inventing land life. They were remembering fragments of it. One gene for UV protection here. One enzyme for osmotic stress there. The building blocks were already present. The transition was a matter of rearrangement and persistence. And all the while, gravity works silently, invisibly. Not just on the body of the organism, but on its internal processes. Fluids no longer distribute evenly. Cytoplasm sags. Nutrients settle. Pressure builds at cell junctions. The development of plasmodesmata channels between plant cells becomes crucial. These structures allow for regulated movement communication, the sharing of signals and sustenance across tissues. In this way, multicellularity is not merely an increase in complexity, but a strategy for withstanding the strain of weight. A single algal filament might collapse under the sun, but a network of cells bonded and communicating can distribute pressure and repair damage. Community at the cellular level becomes survival. There is something gently ironic in how terrestrial life, so often imagined as vigorous, mobile, and expansive, begins as a solution to stillness, as a way to remain, to not be washed away, to resist both motion and decay. The earliest strategies for terrestriality are not movement, but adhesion, not growth, but holding, not breathing, but sealing. These algae drifting into the possibility of land are not explorers in the romantic sense. They are fragments that stay, because staying becomes slightly more possible with each generation. One extra hour of moisture, one additional molecule of shielding pigment, one more layer of cellulose. The mystery that lingers is not whether algae became plants, that lineage is well supported, but why such a transition happened only once. Why among the many photosynthetic lineages that coat the Earth's seas, only a narrow group stepped onto stone and remained? The answer may be biochemical, it may be historical, or it may lie in the unrecorded particulars of a million microhabitats long eroded where survival became slightly easier, slightly more likely for these few. Evolution is not a series of victories, but a long forgetting of failures. What we see is what remained, what endured. So the edge of the land begins to change. Slowly, imperceptibly, it takes on color, not the deep forest green of later centuries, but the faint staining of rock with chlorophyll and its derivatives. A green smear here, a crust there. Algae that darken when dry. Surfaces that shimmer slightly when rehydrated. The signs are easy to miss, and yet they mark a boundary crossed. Not in a leap, but in a million silent adjustments. Not in triumph, but in quiet resilience. Not by choice, but by the slow sculpting of exposure. The algae do not know they are becoming something new. They are only trying not to die. On the margins of the ancient shore, where water recedes and returns without rhythm, a new kind of resilience begins to form, not in stems or leaves which do not yet exist, but in the reproductive whispers of early plant-like algae, the spore. It is small, unassuming, and mostly invisible to time. No roots anchor it. No sunlight warms its path. And yet within its tiny wall lies a memory of wetness, of darkness, of the precise conditions under which life might begin again. These spores are not seeds. They are simpler, more ancient, and far more vulnerable, yet they hold a quiet defiance. To be cast into the air, to land where no tide flows, to wait for moisture, without guarantee, this is not only reproduction. It is an act of faith in absence. It is how life learns to persist where life has not yet been. The spores of these early algal lineages are toughened, layered with sporopollenin, the same stubborn molecule that will one day protect the pollen of forests and flowers. This compound does not decompose easily. It resists heat radiation and the slow abrasion of sediment. Its origin remains uncertain, a molecular adaptation perhaps first meant for UV shielding or microbial defense, later co-opted for reproductive casing. Whatever its path, it becomes the key to terrestrial endurance. In the fossil record, spores often appear before plants themselves, drifting through strata as specks of persistence. Their shapes are often trilet marked by 3 lines that converge at a point, a relic of the tetrad structure from which they split. These marks are not ornamental. They are signatures of a shift in life's strategy, to leave water not as organism, but as potential. Unlike aquatic reproduction, which can rely on liquid to carry gametes and to fuse cells, terrestrial spore dispersal is lonely. Wind replaces current. Gravity replaces buoyancy. Fertility becomes a matter of proximity and patience. Many spores do not germinate. Many dry and vanish. But those that fall into damp crevices, into the shaded corners of rock, or the temporary puddles of rainfall, may split open and begin again. The timing is delicate. The window is narrow. Every successful germination is a minor statistical miracle. And yet the strategy works not because it is efficient, but because it repeats. Again and again spores are cast into uncertainty. Enough succeed to make failure survivable. The poetic irony rests in their scale. These tiny, unnoticeable bodies, smaller than dust, lighter than breath, carry the weight of entire evolutionary futures. They are not complex. They contain no guarantees. But within them are the blueprints of multicellularity, of terrestrial form, of genetic lineages that will one day populate forests and marshes, savannas and tundra. The vast ecosystems of later Earth begin in these fragile ovals, not with grandeur, but with replication. Not with dominance, but with vulnerability dispersed wide enough to matter. Scientists track them through ancient sediments, mapping their densities and patterns, inferring where and when land life began to gain footholds. Trilete spores first appear in the mid-Ordovician sparse and scattered likely from organisms still aquatic, but already experimenting with emergence. By the Silurian, they become more frequent, more varied their walls, thicker, their ornamentation more intricate. These details are not aesthetic. They are functional. The folds and ridges increase surface area, aiding in wind dispersal or adhesion to moist surfaces. Some scientists hypothesize that early spores may have piggybacked on insects or fungal threads, but no consensus has yet crystallized. The mechanisms are hazy, but the presence is clear. Life has begun to project itself into dry space. Mystery lingers in the specifics. What environmental pressures pushed these algae to abandon direct water-bound fertilization? Was it the unpredictability of tides, the competition for light in deeper pools, the scarcity of stable freshwater zones? Perhaps the very irregularity of the shore, sometimes dry, sometimes flooded, selected for those who could hedge against disappearance. The spore is a hedge, a gamble cast wide. It does not seek to control outcomes. It only seeks to increase the possibility of presence to maintain a foothold across time. In this sense, spores are a memory of moisture shaped to survive its loss. There is a gentle, persistent observation to be made here that life expands not through certainty, but through distribution. The spores of early plants do not seek fertile ground. They do not target their landing. They go where wind or water sends them and wait. And in this waiting, they transform how life occupies space. No longer confined to water's edge, organisms can now leap distances, traverse arid stretches, colonize the high edges of rock and the shaded sides of boulders. Each spore is an emissary of survival, carrying not only genetic material, but the possibility of a second chance. The structural evolution accompanying this shift is subtle. Specialized cells begin to form within the algal body, dividing meiotically wrapped in dense walls, released only when dry conditions suggest dispersal may succeed. The body of the plant becomes more than a photosynthetic surface. It becomes a vessel, a protector, a release mechanism. With spores comes a shift in bodily function from simple growth toward temporal strategy, from presence toward reproduction with delay. These are not yet true land plants. There is no root, no shoot, no vascular flow. But there is a before and after. There is a change in how life imagines time. Today, in damp woods and alpine moss beds, spores still drift from liverworts and hornworts, near descendants of these early colonizers. They shimmer in the morning sun, barely visible, curling into hygroscopic shapes that crack and release their contents when humidity drops. Botanists observe their motion under microscopes, tiny springs of desiccated tissue twisting open, launching reproductive particles into air. What was once a molecular adaptation is now a biomechanical performance. But the principle remains to escape the body, to survive in waiting to land where life might begin again. The strategy is unchanged across nearly 500 million years, so the shoreline continues its quiet transition. Spores gather in the cracks of basalt, in the shaded undersides of cobble, in the leeward hollows of fossil dunes. The rock remains unchanged to the eye, but within its microhabitats, something new has begun. Life no longer dies with the tide. It leaves instructions behind, tiny, durable, and unread until rain returns. Each spore a breath held across centuries, each germination a memory rehydrated. And with each cycle, the land listens, learning to cradle green. In the stillness before soil, when land remains bare, and mineral and water clings only in films along stone, life begins to test a new form of alliance, not of kinship, but of cooperation. Among the algae pressing into damp crevices and sending spores into air, and the microscopic fungi threading through cracks and decaying organic traces, there emerges a quiet partnership. It does not begin with recognition. It begins with proximity. Fungal hyphae fine as mist weave through the rock's interstices, drawn by minerals, by carbon, by the faint signals of photosynthetic metabolism. The algae, for their part, produce sugars, products of sun and air, and patience molecules not easily found in lifeless terrain. Where their filaments meet through contact, through chemical gradient, through chance, a mycorrhizal prelude unfolds. The first union of plant and fungus does not resemble the root and branch symbiosis of later forests. It is soft, provisional, a mingling of surfaces, and yet it changes everything. The fungus brings access. Its filaments extend far into the rock, reaching ions the alga cannot touch. Phosphorus, nitrogen, trace metals, these flow along fungal threads toward the photosynthetic body. In return, the fungus receives carbohydrates, simple sugars derived from the sun, carried through cell walls in molecular exchanges, so ancient their mechanisms remain partly unknown. This is not parasitism, nor is it fusion. It is mutualism born from mutual lack. Each organism lacks what the other has found a way to reach, and so together they persist, not stronger, but less vulnerable, not faster, but more stable. In modern biology, these symbioses are studied with fluorescence and isotopic tracing. In greenhouses, spores are cultivated with sterile seedlings, mycorrhizal networks re-established in petri dishes to reveal their functions. But in the Devonian margins, where they first evolve, there are no roots, no soils, no leaves to trace. Only algae holding fast to stone and fungi, seeking decay where none yet exists. Together, they begin to create something new, the earlier soil. Not yet the loam of gardens or the dark beds of forests, but a layer of life-altered mineral etched and softened by acids, threaded with filaments, darkened with carbon, capable of retaining moisture where before it ran off. This process is achingly slow. Rock weathers by frost and heat, by time and tectonic fracture, but with fungal aid, it begins to weather biologically. Organic acids leach silicates, liberate ions, create voids. The fungal networks stabilize fragments, slowing erosion, binding grit into matrix. The algae anchor these protosoils with their own decay cell walls, mucilage pigments, sugars, all feeding microbes whose metabolisms further alter the substrate. Soil in its infancy is a tapestry of partnerships, not merely particles but processes, and beneath it all, the silent presence of fungus, ancestral and unseen. The irony is persistent that before plants could shape the land, they required something not plant. That before roots could drink or stems could rise, a fungus a kingdom apart made it possible. Without this partnership, the transition from algae to land plant would stall at the shoreline. Nutrients would remain locked in stone. Moisture would vanish too quickly. Stability would falter. But with fungal collaboration, the land begins to yield. Not by force, but by dissolution. Not by conquest, but by conversation. Uncertainty surrounds the origins of this relationship. Did fungi arise first on land? Some fossil evidence suggests that soil fungi may have predated terrestrial plants by millions of years. Their hyphae appear in Paleosol's fossil soils long before the earliest vascular stems. Molecular clock estimates drawn from gene divergence hint at ancient lineages spreading across bare continents, seeking carbon in microscopic forms. Whether these early fungi partnered with cyanobacteria with protists or with algae remains unclear. But by the time algae edge toward land, the fungal presence is already there waiting. The match is not orchestrated, it is stumbled into. And it changes the fate of both. Today, almost all terrestrial plants rely on some form of mycorrhizal interaction. In forests, networks of fungal threads connect entire groves, exchanging nutrients, transmitting signals, stabilizing ecosystems. These complex systems, dubbed the Wood Wide Web, trace their ancestry to the humble alliances of algae and fungi on bare stone. The leap from solitary survival to interdependent persistence begins not with intelligence or choice, but with cellular compatibility and biochemical openness. What we now call symbiosis once began as proximity, as experiment, as accident, repeated until it became necessity. There is a poetic symmetry in how life solves its hardest problems, not alone, but together. Photosynthesis does not suffice. Respiration alone is not enough. But together, algae and fungi survive what neither could endure alone. In this early landscape, there is no competition for space, no stratification of canopy and floor. There is only flatness, wetness exposure, and within it pockets of shared endurance. Lichen-like forms appear, though their ancestry is debated. They spread slowly, crusting rocks in thin layers, exchanging sugars and signals resisting desiccation better than algae alone. Whether they are true lichens or ancestral analogues remains a question. But their presence speaks of union. What emerges is not just survival, but transformation. The land begins to retain water. Sediment no longer flows immediately to sea. Microbial populations diversify, drawn to the organic traces left by plant fungal decay. The first stable surface forms capable of hosting more than a single layer of life. The land no longer merely bears organisms, it supports them. And beneath this support lies the hyphae, the net, the silent collaboration that began without notice. So much of this remains invisible. Fossil records capture only impressions, rare mineralized remnants. Mycorrhizal associations do not fossilize well. Instead, scientists infer them from nodules, from preserved root patterns, from isotopic anomalies in ancient soils. Genetic studies confirm the presence of fungal symbiosis genes in the earliest land plant lineages, deeply conserved, strongly expressed. But the original moment, the first cell to exchange sugars with a thread of fungus, is unrecoverable. It lies behind the curtain of time, shaped not by intent, but by compatibility. And so in this quiet phase of Earth's transformation, life takes a step not through mutation alone, but through alliance. The first true greening of land does not begin with forests or ferns. It begins with a whisper between kingdoms, with carbon traded for minerals, with light shared through darkness. It begins with organisms that in their joining reshape stone moisture and time. Along the humid seams of ancient shorelines, in places where mist lingers longer than sunlight, and water pools in shallow depressions carved by time, the algae-fungal unions begin to take new shape. Slowly, almost imperceptibly, the flattened thalli of early liverwort-like organisms emerge. They are not tall, nor dense, nor complex in the way later flora will be. They lack roots, lack true leaves, and grow only a few millimeters thick, but they are something the land has never hosted before plants. Not in the modern sense, not yet, but in structure, in function. In their genetic and cellular alignment, they now carry the unmistakable identity of land-dwelling vegetation. These liverworts are not explorers. They do not march inland with ambition. They press into crevices, cling to damp stones spread across the horizontal world with an almost glacial patience. Their tissues are thin enough to absorb water directly, to draw in gases through every exposed surface. They breathe not through mouths or lungs, but through pores, primitive openings that remain fixed, always ajar, incapable of closing, vulnerable to drying, but essential for gas exchange. In these small patches of tissue, carbon dioxide enters, and the quiet machinery of photosynthesis hums slowly, converting air and sunlight into sugar, leafless and silent. These early plants reproduce with spores carried on small structures that rise slightly above the thallus surface. They lack the elegance of flowers or the architecture of stems, but within their simplicity lies the secret of their persistence. Their gametophytes dominate the life cycle, spreading outward through fragmentation and spore dispersal, sending forward genetic continuity in structures designed for wetness and close contact. Rainfall becomes essential not only for hydration, but for fertilization. Sperm, still motile, still swimming, require a thin film of water to reach the egg. Fertility then depends not on proximity alone, but on weather. A storm can mean lineage, a drought extinction. The land does not welcome life with ease. Every reproductive attempt is a negotiation with dryness, a wager placed on the return of rain. Yet the liverworts endure. They grow flat and wide, maximizing contact with the substrate. Their surfaces shimmer with secretions mucilage that retains moisture, feeding microbial partners, sheltering tissues from rapid desiccation. They adapt to their mineral world with a quiet inventiveness, not by changing the land, but by yielding to its textures. They do not penetrate stone, but conform to it, spreading where water rests longest. In doing so, they extend the reach of photosynthesis across terrain previously untouched. The greening of rock continues no longer just by algae or lichen-like films, but by organized tissues capable of structure and inheritance. Fossils of these plants are rare. Their tissues decay easily, yet their spores persist, tiny ornamented spheres found in ancient sediments, chemically distinct evidence of a terrestrial phase unfolding across millions of years. Molecular data supports their antiquity. DNA analysis of extant liverworts reveals lineages that diverged early basal to the land plant tree, holding within them signatures of ancient adaptations, UV protection, water transport mechanisms, desiccation tolerance. Modern relatives retain these traits. In tropical cloud forests, liverworts blanket tree trunks and rock faces, mirroring the habits of their ancestors, thriving in the narrow ecological window between saturation and sun. The irony is in their modesty. These are not plants that transform landscapes with height or root or flower. They do not sculpt valleys or stabilize soil on a grand scale. Yet they arrive first. They open the land to vegetative life. They soften the interface between biology and geology. They prepare the terrain not by reshaping it, but by surviving it. Through their presence, they invite others' microbes that break down their tissues, fungi that extend their reach, insects that begin to crawl across their surfaces. The land, once sterile and sharp, becomes layered with life. And with them comes a shift in temporality. Before, life was seasonal, only in the ocean's rhythms, in light and dark, in currents and tides. Now life must adapt to longer dry spells, to cycles of moisture measured not in hours, but in days, weeks, years. The liverworts do not resist this change. They dry and rehydrate, shrink and revive. In desiccated form, their cells collapse inward. Metabolism halts, but when water returns, they unfurl, resume photosynthesis, continue growth as if no time has passed. This is not stasis, but resilience. A biological pause button pressed when needed, released when the land permits. There is a quiet mystery in how they manage this. The biochemistry of desiccation tolerance is complex, involving sugars like trehalose proteins that protect membranes, regulatory genes that sense hydration status. Many details remain uncertain. Why some lineages gained these abilities and others did not is not fully known. Why certain proteins persist across vast evolutionary distances, from moss to liverwort to resurrection plants, while others fade from use, remains a subject of study. The liverworts do not offer answers. They only endure. They also blur categories. In their simplicity, they challenge definitions. They are plants, but not vascular. They photosynthesize, but have no true leaves. They spread, but do not grow tall. In the modern taxonomy of flora, they sit uneasily, often overlooked, often underestimated. But in deep time, they are pivotal. Their arrival marks a shift not only in habitat, but in possibility. Before them, land was a place life could touch. After them, it becomes a place life can stay. So the landscape changes once again, not through spectacle, but through accumulation. Liverworts spread in thin mats, darkening stone, trapping silt, slowing runoff. Their presence allows water to linger slightly longer. Their decay provides the first organic input for soil-forming bacteria. The rock begins to hold life not only as visitor, but as resident. From afar, the terrain still appears barren. No trees rise, no canopy forms. But underfoot, at the scale of millimeters, the quiet invasion is underway. Life is not climbing, but crawling. Not reaching, but resting. And in resting, it begins to shape the land with softness, with breath, with silence. The first real plants are here. And they have come not to conquer, but to persist. Across the low damp surfaces where liverworts spread and spores scatter the air, remains an ever-present threat, invisible, weightless, and yet capable of pulling water from tissues with relentless persistence. Desiccation is no longer a passing inconvenience, but a structural challenge. On-land water is not a given. It must be held protected, guarded at every surface. And so quietly, another adaptation begins to form, a thin, waxy layer secreted by cells, a cuticle. It is not armor, not visible to the naked eye, but a microscopic film of lipids and hydrocarbons that alters the relationship between organism and air. Where once cells opened freely to the sky, now they begin to shield themselves. The cuticle is not thick. It is fragile, permeable, in places often patchy. But it reduces evaporation, slows. The loss of moisture extends the hours of activity between rainfall and drying wind. In it, a new kind of threshold is born, one that allows photosynthesis to continue without immediate rehydration, one that buys time in the long contest between water and air. The earliest cuticles are uneven. They do not coat every surface. Their composition varies. In some plants, they contain waxes that reflect sunlight, in others compounds that resist microbial attack. Over time, the cuticle becomes more complex, layered, structured, embedded with pores and channels, but at its origin it is simply a way to remain hydrated for just a little longer. In doing so, it permits everything else. A plant that does not immediately dry can continue to photosynthesize, to grow, to send spores into air with greater reliability. Moisture is no longer a fleeting gift. It becomes a managed resource. In modern plant anatomy, the cuticle is studied through staining electron microscopy chemical assays. Its thickness is measured in micrometers. Its components catalogued: cutin, wax esters, long-chain fatty acids. But its evolutionary emergence is inferred from indirect evidence, from the fossilized impression of early land plant tissues, from the pattern of preservation in spores, from genes conserved across mosses, hornworts and vascular descendants. It is a trait shared widely, though with variation, a sign that it emerged early and was refined often. The exact moment is lost, but its presence marks a turning point. The shift from passive moisture dependence to active moisture control. This innovation carries with it a trade-off. To seal water in is also to seal gases out. Photosynthesis requires carbon dioxide, and gas exchange must occur. The earliest plants solve this dilemma with permanent pores, stomata, that do not open or close, simply gaping pathways where gas can diffuse. These pores likely present in hornworts, and some liverwort lineages permit respiration, but also leak water. In later plants, stomata become regulated, able to close in dryness, open in light pulse with rhythmic cycles. But here, in these earliest experiments, the balance is precarious. The cuticle is not impermeable. The pores are not controlled. Survival depends on microclimate, on shaded crevices, on humid dawns, on the rhythm of mist and drizzle. Each plant clings to its niche. Evolution again does not demand perfection. Only enough improvement to allow persistence. There is irony in the thinness of this adaptation. No more than a few molecular layers, the cuticle is a barrier that defines the future of terrestrial life. Forests, crops, mossy fields all depend on this invisible skin. Without it, water would vanish in minutes. With it, life holds to land. Yet it is not flashy, not sculptural. It does not grow tall, bear seeds, or bloom. It simply coats, preserves, and enables. It allows a plant to remain where otherwise it would vanish. Scientists puzzle over the origin of the biosynthetic pathways involved. The enzymes that construct cutin and wax must be regulated, must assemble molecules precisely. Many of these enzymes appear to be homologous across land plants, suggesting deep ancestry. Some have parallels in aquatic algae used perhaps for defense or flotation. Whether the cuticle evolved once and diversified or multiple times in different lineages remains uncertain. The fossil record cannot resolve the chemistry of these early barriers. It can only suggest function through patterns of preservation and ecological expansion. What is known is that once the cuticle emerges, plants begin to colonize drier spaces. Not arid regions, not yet, but areas exposed to sun between rainfalls, the higher edges of stream banks, the wind-scoured faces of boulders. The cuticle permits risk. It opens access to marginal habitats. Plants can now exist not only in constant wetness, but in places where water arrives intermittently. The land, long inhospitable, begins to offer opportunity. And the plants no longer tethered to every drop begin to spread. Still, the cuticle does not solve every problem. Water must be absorbed, and without roots, absorption occurs through surface contact. The cuticle slows this too. It is a barrier in both directions. A paradox emerges. The very adaptation that preserves water also complicates its uptake. Plants adapt with specialized tissues, with thinner regions, with capillary mechanisms along margins. The balance remains delicate. Evolution continues its adjustments, always partial, never final. In modern ecosystems, we see the legacy of these negotiations. Desert plants thicken their cuticles, add waxes, reflect light. Tropical epiphytes thin theirs, absorb mist directly. Liverworts retain a patchy cuticle, their surfaces still soft, their margins still glistening. Each lineage carries a version of the solution. Each adapts the same problem to different ends. The principle, however, remains: water must be kept, or land cannot be lived upon. And so the cuticle becomes one of the quiet revolutions of terrestrial life. Not a structure one can hold or see, but a shift in the relationship between organism and air. It turns the external world from a solvent into a medium, from an enemy into an element that can be withstood. It permits the separation of life from immediate surroundings. A plant can now experience the environment not as an extension of itself, but as something to be navigated, buffered, negotiated with. This is a subtle, profound change. Before the cuticle, life and water are inseparable. After it, life begins to shape its internal world. The cell is no longer entirely open to the environment. Regulation begins. Homeostasis emerges. Not perfectly, not fully, but enough to matter. Enough to persist across dry nights, across sunny ridges, across brief interruptions in rain. The landscape begins to shift in response. Patches of liverworts, hornworts, early mosses now persist through more variable conditions. They do not climb, but they expand. Their presence weathers rock traps, dust forms the thin beginnings of organic crust. The air begins to change too. As photosynthesis increases, carbon dioxide is drawn down, oxygen is released. The planetary feedback begins slow and steady, and beneath it, the cuticle holding water in the face of the wind, never seen, never named, but essential a skin that allows land to remember moisture. Beneath the soft crust of mosses and liverworts, in the shallow mats of early plants that stretch across damp stone, something shifts in the body's architecture. Until now, water moves only by contact, by surface tension, by the slow diffusion across open cells or narrow filaments. There is no internal transport, no system for moving moisture or nutrients beyond the reach of the nearest drop. But as plants grow more complex, still flat, still close to the ground, yet now broader in surface and longer in form, a new necessity arises: the ability to move water internally from one place to another, regardless of direct exposure. This is not a leap toward complexity, but an accommodation of expansion. And so slowly the first conducting cells appear, not yet vessels. As we know them, not yet xylem or phloem, but elongated cells with walls that guide flow channels, where water can pass from base to tip, from shaded root-like filaments to sunlit surfaces above. The movement is passive, drawn by evaporation at the surface, pulled by the gentle suction of transpiration through pores. It is not fast, it is not strong, but it is direction. And direction means growth. These primitive conduits are called hydroids, long, thin, water-carrying cells found in the stems of early mosses and their relatives. Their walls are thin, their structure delicate, yet they represent a fundamental reorganization. Water no longer surrounds the organism entirely. Now it flows through it. Nutrients too can be pulled from the soil, if soil there is, or from the damp crevices between rock grains and transported upward. This is no longer a mere mat of cells photosynthesizing in place. This is a body with gradient, with flow, with internal polarity. With vessels, however primitive, comes verticality, not in stature, not yet, but in function. The irony is gentle and persistent. The taller a plant grows, the harder it becomes to hydrate. To reach even a few centimeters into air requires a strategy to prevent cells from drying, to ensure that moisture and dissolved minerals arrive where needed. The internalization of water movement solves this problem incrementally. Each new adaptation, slightly wider hydroids, slightly more lignified walls, slightly longer conduction pathways, permits slightly greater height. There is no rush toward trees, no intention of canopy. But with every improvement in internal transport, the land gains a new dimension of life. The fossil record offers glimpses. In early Devonian rocks, fragments of stem-like structures appear with central tissues, suggestive of primitive vascular bundles. Some contain cells with thickened walls, laid down in rings, or spirals patterns that resist collapse under pressure. These are the precursors of tracheids, the first true vascular conduits. But even before these appear, the language of vessels is already spoken quietly in the damp lowlands, where mosses spread and spore capsules rise. It is not a new language, but an old one spoken in new terrain: the movement of fluid, the preservation of turgor, the distribution of solutes, all reorganized for a body that lives in contact with air. Modern analogues provide clues. In the moss, Polytrichum for instance, water moves through a central core of hydroids surrounded by leptoids that carry sugars. This arrangement is not homologous to the xylem and phloem of vascular plants, but it performs a similar function. The distinction between these tissues and those of higher plants is significant, but the principle is shared structure for flow, internal space, repurposed for motion. Even the simplest conduction cell must resist collapse, must permit water to pass without leakage, must integrate into the plant's architecture without weakening its body. These demands drive cellular innovation. Wall-thickening lignin precursors program cell death to remove internal contents and allow fluid continuity. Mystery surrounds the origins of these systems. Did vascular tissues evolve once in a common ancestor of all tracheophytes, or multiple times in different lineages under similar pressures? Are the conduction cells of mosses a parallel invention, a separate solution to the same problem? Genetic analysis reveals partial homologies: genes involved in cell wall patterning, in lignification, in tissue differentiation. Some are ancient, shared across plant groups. Others are more derived, restricted to specific clades. The evolutionary history is not linear, not simple. Like the vessels themselves, it winds through many channels. And with vessels comes change beyond the plant. As early vascular tissues evolve, as more water is drawn from the soil and moved upward, the surrounding rock begins to change too. Increased uptake leads to greater weathering. Minerals are pulled from stone and incorporated into biomass. When plants die, these elements are returned to the land in altered form, organic bound, partially decomposed, chemically distinct. Soil deepens. Horizons form. The land becomes layered not just with life but with its residues. The vessel is not merely a tool for growth, it is a vector for transformation. The ecological consequences extend outward. As plants become better at moving water, they can survive in drier microhabitats. A slope that once supported only algae may now host a patch of moss. A surface exposed to occasional mist becomes habitable. Vegetation spreads in patchwork, following the contours of moisture availability, but no longer confined solely to places of constant wetness. The vessel extends the reach of life. It does not guarantee success, but it shifts the limits. There is a soft irony in the comparison to animal systems. Blood vessels, too, move fluids internally. They carry oxygen nutrients waste. Yet where animals rely on muscular pumps, plants rely on physics, on pressure differences, on capillary action, on the cohesion of water molecules. The plant's solution is quieter, slower, but no less effective. It allows them to remain rooted, unmoving, yet connected internally. The stillness of the plant is not inertness. It is a different kind of motion, one that happens within. So the land greens not only in surface, but in structure. The mats and thalli of earlier forms give way to filaments and stalks, to cushions and tufts, to bodies, with central strands and peripheral tissues. These are not yet ferns, not yet shrubs, but they are no longer just algae with attachments. They are organisms, with direction, with tension, with inner flow, and with every drop of water lifted upward, they reshape what land life can be. Beneath the fragile green that spreads across the margins of stone and sediment, life begins to test the idea of anchorage. The early land plants, mosses, liverworts, and their vascular kin to bee cling where they can, but their hold on the land remains precarious. Wind can tear, rain can wash, and desiccation still haunts every inch of upward growth. What they require now is not only the ability to stay hydrated or to transport water, but to remain, to resist removal, to endure motion, to attach with persistence deeper than mere contact. And so they begin to reach downward, not with true roots, not with the specialized organs that will later burrow meters into soil, but with rhizoids, simple hair-like filaments, delicate threads that anchor without absorbing that grip, the microscopic contours of stone and sediment like a memory of descent. These rhizoids are not yet roots, but they are shadows of what roots will become. They arise from individual cells extending into crevices, wrapping around grains of sand, spreading in mats so fine they blur the line between plant and substrate. Some are unicellular, others are multicellular depending on the lineage. They do not branch, they do not grow deep, but they hold. And in holding, they allow the plant to face the world, not just with its surface, but with an orientation upward for light, downward for place. This new polarity is not spatial alone, it is functional. Plants now have a top and a bottom, a source and a sink, a path for movement and exchange. Even without true roots, they begin to occupy land with intention. The emergence of rhizoids does more than prevent dislodgement. It opens a new phase in the relationship between plant and substrate. Rhizoids stabilize soil, or the primitive equivalent of soil binding particles together, preventing erosion, encouraging the accumulation of organic matter. As plants grow and die, their tissues feed microbial communities that further break down rock, increasing the mineral availability of the immediate surroundings. Over time, the spaces between stone and plant become richer, moister, more structured. Rhizoids trace these microhabitats, following gradients of water temperature and chemistry, extending not just reach, but presence. Fossil evidence for rhizoids is elusive. Their softness does not preserve well, and their microscopic scale makes them difficult to detect. Yet impressions in fine sediment, subtle alignments of grains in ancient siltstone, and the occasional fossilized base of early plants suggest their presence. More compelling is the genetic legacy they leave behind. The molecular machinery required to produce rhizoids is conserved across plant lineages. Transcription factors signaling pathways, cell wall enzymes. These are not unique to modern mosses or ferns. They are ancient, shared, indicative of a trait that evolved early and proved indispensable. The irony lies in their simplicity. Rhizoids are not vascular. They do not conduct water in bulk. They do not actively seek out nutrients. And yet without them, early plants could not have maintained their place on land. To grow even slightly above the surface requires a counterbalance, a way to stay in one spot long enough to complete the cycle of reproduction of spore release and germination. Rhizoids provide this quiet resistance. They root the plant, not in the way a tree anchors with vast branching systems, but in the way a breath roots a body in stillness, gently, pervasively enough. Their origin remains partly mysterious. Some algae produce rhizoid-like structures used to attach to underwater surfaces. Whether the rhizoids of early land plants evolved independently or represent a modification of these ancestral traits is debated. Genetic evidence suggests both continuity and innovation. What is clear is that rhizoids were present in the earliest land plant lineages, and that they represent one of the first true terrestrial adaptations focused on position rather than hydration. In modern ecosystems, the legacy of rhizoids continues. Mosses spread across tree trunks, their rhizoids clinging to bark and stone. Liverworts sprawl across damp soil, held in place by invisible threads that resist gravity and rain. In peatlands, entire landscapes are built upon generations of rhizoid-bound plants, forming spongy masses that store vast quantities of carbon. Though often overlooked, rhizoids are part of the quiet scaffolding of terrestrial life. They do not rise, they do not move, but they enable both, and they offer something else, a medium for microbial interaction. Bacteria, archaea, and fungi colonize rhizoid surfaces, drawn by exudates, by moisture, by the steady presence of living tissue. These early interactions may not have been as intimate as mycorrhizal symbioses, but they represent the beginning of another partnership, one in which the plant provides habitat and carbon, and the microbial community returns nutrients and resilience. Over evolutionary time, these exchanges deepen. Roots will eventually become the nexus of soil ecosystems, but rhizoids are the quiet precursors, the test run of below-ground connection. Their structure is modest yet effective. Unlike roots, they do not branch in complex ways, do not form root hairs, do not engage in secondary growth. But they can form dense mats, covering large areas, locking in place the thin films of early soil. When seen under magnification, they reveal adaptations suited to survival in unstable conditions. Roughened surfaces for grip, elastic cell walls for tension, and in some species, the ability to regrow rapidly after disturbance. They are not inert. They respond to touch, to humidity, to nutrient gradients. They are rudimentary sensors, extensions of awareness into the substrate. As the first vascular plants begin to evolve with their own primitive roots, branching from embryonic stems, rhizoids begin to fade from prominence. But they do not disappear. They persist in bryophytes and gametophyte generations, in non-vascular flora that continues to thrive in environments too extreme for larger plants. And so the rhizoid remains both as a living structure and as a metaphor, a reminder that attachment precedes exploration. That presence requires grip, that even the most advanced life forms must begin by holding on. So the land continues to green, not in bursts of canopy, but in layers of thin persistence. The sky remains open. The soil remains shallow. But across rocks and slopes, in moist valleys and high plateaus, plants are now not only growing but staying. Their rhizoids stretch quietly into the earth, forming no architecture, leaving few traces, but carrying forward a single essential lesson: to live on land is to let go of water and reach for stone, not with strength, but with persistence. There comes a point somewhere in the shallow basins and shadowed ledges of the Silurian and early Devonian landscapes, when green no longer exists only in isolated streaks along stone or in patches of damp shadow, but begins to form a continuous fabric, a living carpet stretching across wide stretches of terrain. These are not forests in the modern sense. There are no trunks, no branches, no canopies to stir in wind. But they are forests by another measure. Expansive, persistent photosynthetic communities that cover the land's surface layered and self-perpetuating, a low world of liverworts, mosses, and early hornworts that blanket rock and sediment like breath held close to the earth. They are short, often no taller than a fingernail, but they are vast. Where water lingers after rain, where mist rolls down from uplands, these kneeling forests stretch in dense, humid silence, their surfaces wet with morning dew and shaded beneath their own minimal canopy. The irony is immediate and enduring that the first forests grow, not by reaching upward, but by spreading outward, not in pursuit of height, but in the conservation of moisture and surface contact. Every millimeter of vertical growth increases the challenge of hydration. Every new layer of tissue must be justified by an equal increase in structural support and internal transport. But in this low sprawl, such challenges are softened. Light is absorbed efficiently by a flat, dense mat. Water is retained beneath overlapping tissues. Reproduction is aided by proximity. Spores released from one gametophyte often land within reach of another. Fertilization, still dependent on liquid, becomes more likely when the community is dense and damp. This horizontal strategy, while limiting in stature, maximizes continuity. The result is a terrestrial biosphere that, while miniature, is stable, connected, and ecologically active. These early plant communities create conditions previously unknown on land. They intercept rainfall, slow runoff, and allow water to soak into cracks and fissures. In doing so, they reduce erosion, retain nutrients, and foster microenvironments for other life. Cyanobacteria, fungi, protists, and early invertebrates now find shelter and sustenance among their tissues. A thin boundary layer of humid air forms just above the mat, reducing evaporation and buffering temperature extremes. This microclimate, mere centimeters thick, allows for persistence in places where exposed stone would otherwise remain lifeless. The landscape begins to breathe in new ways. Gases are exchanged not only between ocean and atmosphere, but across every square meter of moss-draped land. Fossil evidence supports this transformation. In certain Devonian deposits, preserved mats of early plants appear as dark compressions in shale and siltstone, sometimes accompanied by spores, fragments of thalli, or impressions of flattened stems. Some show evidence of decay and regrowth, signs of seasonal cycles of disturbance and recovery. Others are layered, indicating slow accumulation over time. These are the archives of the first stable terrestrial ecosystems, built not on height, but on resilience, not on innovation alone, but on repetition. The fossil record tells us little about their color, their scent, their tactile presence, but the density and persistence of their remains suggests something that held on long enough to matter. There is quiet poetry in the paradox that the earliest plant ecosystems thrive not by dominating the vertical axis, but by refusing it. They invest not in rise, but in reach. Their power lies in continuity, in covering the land so completely that each drop of water finds its place, that each pore has a neighbor, that each loss can be absorbed by a neighboring gain. This is not a strategy born of weakness. It is the first successful response to the total exposure of land to its winds, its heat, its dryness, its unpredictability. To kneel here is not to submit. It is to survive. Modern analogues remain. In Arctic tundra, alpine moorlands and shaded forest floors, mosses still form dense, low-lying carpets that dominate where vascular plants cannot. They slow snowmelt, insulate soil, and store moisture in environments too harsh for taller flora. These are not remnants of a bygone age, but refined continuations of a strategy that has never ceased to be effective. In such places, one sees the living memory of those first forests, not in structure, but in function. And with these mats comes an increased cycling of carbon. As photosynthesis proceeds across millions of square kilometers of low vegetation, Carbon dioxide is drawn from the atmosphere, stored temporarily in tissue, and returned through respiration and decay. Over time, this cycling begins to influence climate. Atmospheric composition shifts. The balance of gases tilts slowly, imperceptibly towards something new. Life is no longer simply responding to environment. It is beginning to shape it, not dramatically, not suddenly, but persistently. There is scientific debate over how quickly these early plant communities altered Earth's systems. Did they draw down enough carbon dioxide to tip the planet toward glaciation? Did their decay feed soil, microbial communities that released methane or stabilized nutrients? The evidence is incomplete, the feedback loops complex, but the trend is clear. With persistent land cover, planetary feedback begins. The biosphere is no longer confined to the sea. The surface of the continents begins to participate in the great cycles of air and water, carbon and oxygen erosion and sedimentation. Yet even now, the plants themselves remain modest. They do not yet produce wood. They do not form vascular cambium or secondary growth. Their bodies remain small, their structures soft, their lives brief and vulnerable. They survive through number, through distribution, through adaptability. Where one patch dies in drought, another thrives in shade. Where one is washed away, spores find purchase downstream. The forest regenerates not from trunk or seed, but from thread and spore, from fragment and pore. And so the first forests of Earth kneel. They hold close to the surface. They press into its textures. They form a skin of living green, where once there was only mineral grey and brown. Their silence is total. They make no sound in wind, no creak of bough or rustle of canopy. They shift only slightly, glistening in dew, rising minutely after rain. Yet in their stillness, they begin to remake the land 1 millimeter at a time. Where once the continents were sculpted only by wind and water, heat and pressure, now the touch of life begins to leave its own impression. The surface, once bare and silent, is slowly altered by the presence of organisms that not only grow upon it, but feed into it, decay upon it, and alter its chemistry with every breath and spore. As the first mats of mosses, liverworts and hornworts blanket the land in low green expanses, they begin quite unintentionally to rewrite the nature of stone itself. This change is not sudden. It is not visible from afar. But in the thin layer where biology and geology meet, a transformation begins. Rock for the first time starts to become soil. The process begins at the smallest scale. The plants cling to surfaces, their rhizoids pressing into the microscopic pores and cracks of mineral faces. These threads do not break rock with force, but with time. They secrete organic acids, mild but persistent, that leach cations from the stone, solubilizing phosphorus, potassium, calcium. These acids are not invented for this purpose. They are byproducts of metabolism, exudates from cell walls, but they act upon the substrate with geological consequence. Over time, the topmost microns of a rock weaken, crystals dissolve, surfaces roughen, minerals fragment, and into these new spaces, life pushes further. Dead plant matter accumulates. Tiny fragments of thallus, desiccated spores, degraded pigments, polysaccharide secretions, all fall into crevices, soak into cracks, and decay. Bacteria follow. Fungi spread. Each adds to the chemical complexity of the interface. Decomposition releases carbon compounds, nitrogen species, trace elements. These compounds bind to mineral surfaces, coat particles, form colloids. The result is something that is not quite organic, not quite mineral, a transitional material rich in potential, low in structure. It retains water better than bare rock. It supports microbial communities. It hosts life beyond the plant that initiated it. It is the first soil, not yet dark, not yet thick, not yet layered into horizons. This proto-soil is a veneer, a skin that softens the face of stone. Its formation takes centuries, millennia, but it persists, and with time, it deepens. Each generation of plants contributes to its mass. Each cycle of rain and drought, heat and frost churns and breaks it further. The soil begins to develop texture, porosity, nutrient gradients. It begins to behave not as dead matter, but as a medium one that holds water, binds, carbon filters, air anchors life. The irony is subtle that rock, long, the symbol of permanence, becomes mutable only through the ephemeral bodies of plants that last mere weeks or months. What geology cannot accomplish alone, the breakdown of granite to clay, of feldspar to silt biology, achieves with soft tissues and slow decay. The more fragile the organism, the more profound its effect. There is no chisel, no blast, only the endless repetition of attachment secretion, death and regrowth. The land yields not to force, but to persistence. Scientific evidence of this transition lies in the mineralogy of ancient soils, paleosols, found in the fossil record, showing features like pitting leaching profiles, iron oxidation bands, and microstructures suggestive of biological influence. These soils are rare, preserved only under specific conditions, but they mark a shift in Earth's surface dynamics. Before, sediments were products of physical weathering and chemical dissolution alone. Now they begin to carry biological signatures, isotopic ratios altered by metabolism, organic residues, trace fossils of roots and filaments. The land no longer just receives sediment, it begins to create it. This creation is not isolated. It feeds back into the biosphere. Soil retains water longer than stone. It buffers temperature fluctuations. It concentrates nutrients that would otherwise wash away. With soil, plants can grow in places previously too dry, too poor, too unstable. With soil, microbial life diversifies. Fungi proliferate, bacteria specialize, microfauna emerge. Earth's surface becomes not just habitable but generative. There is mystery in how quickly this change propagated. Did entire landscapes become soiled over a few million years? Or was it slower, patchier, governed by climate, rock type, and biological distribution? Did certain plants accelerate the process? Those with more extensive rhizoids, more acidic secretions, more durable decay products? Or was the shift universal, driven by the fundamental chemistry of photosynthesis and respiration? The fossil record does not answer with certainty. It offers only fragments, a patch of altered shale here, a weathered sandstone horizon there, spores embedded in layered silt. But the trend is clear. By the mid-Devonian, soils are present across much of the vegetated Earth. And with soil comes the storage of carbon. Organic matter binds with clay particles, is protected from rapid decay, is buried beneath new layers. The carbon fixed by photosynthesis is no longer returned immediately to the air. Some of it remains below in darkening profiles, in deepening bands of humus-like residue. Over time, this sequestration affects the atmosphere. Greenhouse gases decline. Global temperatures shift. Climate cools. The feedback is complex, involving ocean chemistry, weathering rates, tectonic uplift. But the role of terrestrial biomass and soil formation is central. Life, having once depended entirely on climate, now begins to shape it. The soil also remembers. In its layers lie the histories of drought and flood, of colonization and retreat. Pollen grains, charcoal flecks, mineral grains worn smooth by ancient winds, all accumulate, stratify, and hold the memory of what lived and died. Each centimeter holds thousands of years. Each profile tells a story written in roots and rot, in minerals altered and elements moved. Long before the first books, long before the first carved stones or etched symbols, the land begins to record. There is something deeply quiet in this change. Soil does not announce itself. It spreads unnoticed forms beneath the body, grows in darkness. And yet without it, the history of life on land would remain shallow. There would be no depth to hold trees, no cushion for seeds, no place for burrowing animals or fungal webs. Soil is not a background. It is a stage, an actor, and an archive. Its origin in these early days is not grand or sudden. It begins with liverworts on rock, with moss decay in puddles, with the secretion of a single acid molecule against quartz. It begins when stone begins to forget that it was once untouchable. As the earliest plants spread across the continents in soft mats and kneeling carpets, the atmosphere begins to change. Invisible to the eye, slow beyond ordinary perception, carbon dioxide, once dominant in Earth's sky, begins to decline, not through catastrophe, not through sudden geologic events, but through the quiet breathing of countless fragile organisms. Each tiny thallus, each spore-bearing stalk, each patch of moss clinging to stone absorbs carbon from the air with every act of photosynthesis. What begins as an individual transaction between plant and light becomes across millions of square kilometers a planetary transformation. Carbon once adrift in the atmosphere is drawn down into living tissues, into decay, into soil. The change is subtle at first. A few parts per million spread across centuries, but the land's greening accelerates the process, and the Earth's climate begins to shift. The mechanism is neither new nor complex. Photosynthesis has always captured carbon using light energy to bind it into sugars and biomass. But for most of Earth's history, this occurred in water in oceans and lakes, where the carbon cycle was buffered by deep reservoirs, where respiration and burial maintained a delicate balance. On land, this balance tips. The biomass produced is more likely to be buried in soil, where decomposition is slower. Organic matter accumulates. Weathering of silicate rocks, enhanced by plant root secretions, and microbial activity consumes additional carbon dioxide from the air, locking it into bicarbonates that wash into rivers and eventually precipitate as carbonates in marine sediments. It is not just life that draws down carbon. It is life interacting with stone, with rain, with gravity, a cascade of small effects amplified by persistence. Fossil soils or paleosols provide evidence of this shift. Geochemical analyses reveal changes in carbon isotope ratios in iron oxidation states in clay mineralogy, all signs that biological weathering and carbon burial were underway. In some of the oldest well-preserved profiles dating to the early Devonian, scientists find significant depletion of atmospheric carbon dioxide inferred from the structure and chemistry of ancient plant-covered surfaces. Modeling studies suggest that the rise of land plants may have triggered a series of climate feedbacks culminating in global cooling events. One such event, the late Devonian glaciation, aligns temporally with the proliferation of vascular plant lineages and the deepening of soils. It is a strange irony that mosses, ferns, and simple rootless plants organisms with no intention, no foresight, help set the stage for ice. This irony runs deep. The fragility of these plants contrasts with the strength of their cumulative impact. They cannot walk, flee, or resist change. They dry, collapse, regrow. Yet together they alter the gas composition of an entire planet. There is no design, no plan. The drawdown of carbon is not a goal. It is an outcome of survival, a byproduct of breathing and growing and dying on land. Evolution selects for persistence in place for reproductive success in drying air, and in doing so selects also for global change. A gentle mystery lies in the exact tempo of this transition. Did carbon dioxide decline steadily over tens of millions of years, or did pulses of plant colonization trigger rapid drops? Were there regional differences with some landmasses greening faster than others? The fossil record is incomplete, and climate proxies are often difficult to interpret. Ice core data does not reach so far back. Instead, scientists turn to rock chemistry, to isotopic shifts, to modeling constrained by fragmentary data. Each layer of evidence is tentative, each conclusion provisional. But the trend is unmistakable. Life alters air. Green reaches into blue and changes it. And with this shift comes a reorganization of Earth's energy balance. Less carbon dioxide means less greenhouse effect. Global temperatures cool. Weather patterns change. The equator remains wet, but higher latitudes begin to see frost, then snow, then glacial accumulation. Sea levels fall as water is sequestered in ice. New habitats emerge along exposed shorelines. The expansion of land plants may not have caused these changes alone, but they participated, accelerated, stabilized. What was once a mineral world ruled by tectonics and tide becomes a biosphere shaped by metabolism and decay. Still, the plants themselves remain fragile. Their bodies are soft, easily crushed, easily desiccated. They have no bark, no seeds, no vascular strength to rise far above the ground. Yet their spores spread. Their mats regenerate. They respond to environmental gradients with uncanny adaptability. Some thrive in cold, others in heat, some in shade, others in wind-exposed ledges. They persist in diversity. It is this diversity, this decentralization, that makes their impact durable. No single species carries the burden. The biosphere spreads, risk adapts, in aggregate survives change by being many and low. There is another layer to this transformation, the change in light itself. As atmospheric carbon dioxide decreases and as oxygen increases through photosynthesis, ultraviolet radiation is filtered differently. The composition of the stratosphere alters, the ozone layer forms more stably, further reducing the threat of DNA-damaging rays. The land becomes more hospitable, not only through water retention and temperature regulation, but through shielding from radiation. The sky itself is changed by what grows beneath it. A cycle of mutual accommodation begins. Plants change the air. The air allows more plants. In modern landscapes, echoes of this quiet revolution persist. Peat bogs sequester carbon accumulated over millennia of moss growth and decay. Grasslands store carbon in roots and rhizomes. Forests fix gigatons each year. The carbon cycle remains dynamic, but its terrestrial component, once absent, is now dominant. Every breath we take carries carbon once fixed by a leaf. Every stone weathered by root and fungus carries the imprint of this ancient shift. So the land settles into a new equilibrium, not static, but stabilized. The biosphere becomes a regulator, not merely a responder. And in this regulation, a new kind of continuity emerges. Planetary cycles become linked to biological ones. Geology becomes biological history in motion. The atmosphere becomes a reflection of leaf and pore, of rhizoid and decay. And all of it begins with the smallest forms, kneeling close to stone holding carbon, not with intent but with presence. Across the moist and carbon-rich terrain of the early Devonian world, plants begin to change their posture. What was once soft and horizontal, a sprawl of thalli, a carpet of spores and rhizoids pressed to rock, begins to edge upward, not in haste, not in great leaps, but in subtle elongations, in slow vertical reach, in the tentative rise of stalks that resist collapse. For the first time in Earth's long history, life explores height not as a strategy of escape, but as a new frontier of light. Yet to rise means to bear weight, to resist gravity, to withstand the stresses of wind and desiccation. And so within these early vertical bodies, a biochemical innovation appears: lignin. This complex polymer deposited into cell walls stiffens and strengthens the tissues it permeates, transforming delicate cells into scaffolded supports. Where once plants yielded to pressure, they now hold form. The first lignified structures are not large. They appear in the axes of simple branching plants, in the sporophyte stalks of ancient bryophyte-like organisms, in primitive vascular tissues that begin to channel water with more force. These are the precursors of xylem dead hollow cells, with walls thickened by spiral or reticulate bands of lignin forming conduits that conduct water upward and resist internal collapse. The benefit is immediate. Taller structures can support their own weight, can lift photosynthetic tissue into brighter air, can release spores from higher elevations, allowing broader dispersal across an increasingly vegetated land. But with this strength comes a cost. Lignin is difficult to produce. It requires energy, enzymatic precision, and molecular coordination. And once in place, it cannot be undone. The cells it fortifies are dead sealed off from metabolism, repurposed into architecture. In these early stages, every lignified stalk is a choice growth traded for permanence. Fossils record this shift in silhouette. Rhynia and its kin, simple branching leafless plants, leave impressions in cherts and siltstones, their vertical stems preserved with vascular cores, their outer layers thickened and reinforced. They are not trees. They have no wood, no bark, no leaves in the modern sense, but they stand. A few centimeters tall, perhaps more. Enough to rise above the mosses, enough to cast shadow. Enough to mark the beginning of vertical complexity on land. And with height comes new challenges: exposure to wind, greater reliance on internal transport, the need for anchorage beyond surface rhizoids. Roots, too, begin to evolve not merely as anchors, but as absorptive organs capable of branching, of exploring volume beneath the surface, of forming mycorrhizal associations deep in the dark. The irony is persistent that the path to height requires death within. Xylem cells die to become vessels. They empty themselves of content, harden their walls, and form pipelines through which life flows. The tissue that allows growth is made of corpses, their architecture preserved for function. In this, the plant body becomes a mosaic of living and non-living parts, a balance between metabolic activity and structural endurance. The more a plant rises, the more it must rely on tissues that no longer participate in the processes of life. Height demands sacrifice. Scientists debate the evolutionary origin of lignin. Was it first a defense against microbes, later repurposed for support? Did its biosynthetic pathway arise from precursors used in cell wall reinforcement, or from pigmentation, or both? Comparative genomics reveals fragments of the lignin pathway in some algal relatives, but the full suite appears only in land plants. Its complexity suggests a long evolutionary tail: gene duplications, pathway integrations, regulatory networks. It is not a sudden invention. It is an accumulation, a reassembly of ancestral components into a function that enables stature. With lignin and vascular tissues comes a new kind of possibility: form. The plant body, once a formless sheet or soft tuft, begins to specialize. Axes and nodes emerge. Branches divide and redivide. Apical meristems direct growth in specific directions. The body becomes patterned not randomly, but under the guidance of genes responsive to light, gravity, and internal signals. What was once a mass of similar cells becomes a hierarchy: tissue types, organs, systems. Leaves will follow, but first comes the ability to hold shape. In modern descendants, we see the continuation of these first steps. Ferns, horsetails, and club mosses, though ancient in origin, still rely on lignified tissues to stand, to conduct, to persist. Trees build on the same principles, scaling them to meters and centuries. Wood at its core is lignin-patterned, layered, organized into annual rings, but chemically descended from the same polymer that stiffened the first vertical shoots. Even in flowers, in herbs and grasses, lignin remains. It fortifies stems, lines, vessels, protects seeds. It is not visible, not fragrant, not soft, but it is everywhere beneath the surface, shaping form by resisting collapse. There is something quietly poetic in this transition. Plants do not rise to flee or to dominate. They rise to reach the sun more fully, to disperse more broadly, to hold space against gravity. Their stillness is not passivity, but resolution. The stiffer their bodies become, the more intricately they must coordinate growth and support. They invent no bones, no muscles, no nervous systems. They build with water and wall with dead cells, and lignified tubes with internal pressure and cell wall tension. Their strength is architectural, not muscular. Their resilience mechanical, not behavioral. And so the silhouette of the land changes. Where once only mosses clung, now thin stems rise. Shadows lengthen. Air flows differently. Spores fall from higher points, carried further by breeze. Light strikes lower layers more obliquely, creating gradients that influence the distribution of later arrivals. Ecosystems begin to stratify, not yet forests, but the beginnings of layered plant life, a three-dimensional biosphere slowly emerging from the flatness of its predecessors. The world takes notice not through vision, but through consequence. The carbon cycle shifts again as lignified tissues decompose more slowly. Oxygen levels rise, fueled by expanding photosynthetic surface. Soils deepen, enriched by the woody debris of upright plants. The biosphere's influence on the lithosphere grows stronger. What began as a green film becomes a green architecture, and at the heart of it, a molecule, lignin, silent and stubborn, encoding the stiffness that allows form to persist. As Earth's tectonic plates drift toward one another, assembling the bulk of the land into a vast singular mass, a new landscape stretches across the equatorial and temperate zones, a supercontinent of ancient stone, lifted and exposed, weathered by time and wind. On this broad canvas, the greening of land begins to scale beyond local carpets and coastal fringes. No longer confined to margins or moist basins, plants now advance inland, threading through river valleys, spreading across floodplains, clinging to upland ledges, where fog gathers and mist lingers in the morning hours. The process is not abrupt. It moves as spores move, as rhizoids cling, as lignified stems rise modestly above mossy ground. But it is relentless. With every generation, vegetation extends its reach, settling into microhabitats once untouched by life. Over time, the interior of the continent, once a silence of rock and dust, becomes a matrix of photosynthetic activity. This spread is driven not by conquest, but by accommodation. As plants develop the capacity to retain water, to transport it internally, to anchor more deeply and withstand desiccation, they become less dependent on continuous moisture. Some begin to tolerate brief dry spells, others grow in thin soil crusts or in seasonally flooded regions. Those with primitive roots probe deeper, drawing moisture from beneath the surface, while others refine their cuticles, protecting delicate tissues from wind and sun. Vascular networks, still simple, extend the metabolic reach of each plant beyond the limits of immediate contact. The result is a biological persistence that can endure fluctuations in humidity, in light, in stability. Each trait incrementally increases the potential for terrestrial residency. Each adaptation, however small, contributes to the widening of the green frontier. The fossil record preserves this shift in fragmentary mosaics, spores embedded in inland siltstones, impressions of axial stems in the margins of ancient braided rivers, root-like structures preserved in floodplain clays. These signs are indirect but cumulative. They tell of plants that no longer hug the ocean's edge, but venture into the interior, occupying spaces once reserved for wind and dust. The supercontinent, likely Gondwana, with pieces of Laurentia and Baltica near the equator, becomes not just a geologic entity, but an ecological one. Its rivers darken with tannins, its rocks wear down beneath mats of liverworts and mosses. Its lowlands become sites of carbon fixation and soil formation. There is irony here, as always. The most expansive greening in Earth's history is accomplished not by towering flora or complex ecosystems, but by organisms no taller than a child's hand. Their height is modest, but their numbers are vast. Their communities stretch unbroken for kilometers, limited only by barriers of climate or terrain. What they lack in size, they make up in continuity. And this continuity has consequences. As plants spread, they stabilize the land surface, reducing sediment runoff, moderating stream flow, and allowing deeper weathering of rock. This in turn enhances the drawdown of atmospheric carbon dioxide through chemical weathering, further cooling the planet, further inviting the spread of plant life. Climate models suggest that this coupling between vegetation and atmosphere creates a self-reinforcing loop. As plants colonize more land, they increase the rate at which silicate rocks weather a process that consumes CO2 and leads to long-term climatic cooling. Lower temperatures and changing precipitation patterns then create new niches, allowing plants to migrate even further. It is not a fast feedback, but over millions of years, it becomes profound. Glacial episodes follow. Sea levels drop. Continental interiors become more habitable. Life, by changing the land, reshapes the sky. Yet the plants themselves remain simple. There are no seeds, no flowers. Leaves are rare, undeveloped. Most have dichotomous branching, where stems split evenly into two, forming networks of Y-shaped axes. Their bodies are small, but their life cycles are robust. Spores protected by thick walls of sporopollenin are released from elevated sporangia, catching the wind and traveling long distances. Fertilization remains water-dependent, but as spores settle into moist microclimates, their progeny root and grow, reinforcing the advance. In this way, land becomes a patchwork of green, some areas lush, others sparse, all subject to the vagaries of terrain and weather. The extent of this early terrestrial expansion is still debated. Some scientists argue for a rapid spread of vascular plants by the late Silurian. Others suggest a slower, more fragmented colonization, with many niches still unoccupied well into the Devonian. The truth likely lies in between a mix of explosive local growth and cautious continental creep, governed by local geology and global climate. What is clear, however, is that by the Middle Devonian land is no longer a lifeless platform. It breathes, weeps moisture, hosts food webs. It is inhabited not just at the surface, but below and above in soil, in stem, in the spaces between spore and spore. And with this inhabitation comes transformation. Landscapes that were once dominated by physical processes, erosion, sedimentation, volcanism, now respond to biology. Slopes retain their form longer. Rivers meander more slowly. Floods deposit finer sediment as vegetation traps silt. A new style of landscape emerges, one that holds its shape, that records its own change. The biosphere and geosphere are now intertwined. Modern analogues for this stage are rare, but hints remain in alpine tundras, where low-lying plants form dense cushions across thin soils, or in volcanic plains where early colonizers grip ash and pumice with root-like filaments. In such places, the green does not dominate by height, but by density and resilience. These systems remind us that complex ecosystems need not tower. They can sprawl. They can persist. They can alter worlds by doing little more than staying where nothing once did. And so, under the sun of a cooling world, with glaciers creeping along the poles and rain falling across once dry interiors, the supercontinent greens. Not in bursts, but in waves. Not in forest, but in field. From the shores inward, from the valleys upward, from the wettest ravines to the driest plateaus that still hold morning dew, plants remake the face of the earth. There are no witnesses but the wind and the rock. No sound but the hush of spores release the whisper of stems bending the near-silent hum of photosynthesis against stone. By the late Devonian, the land breathes in rhythms it did not once know, a breath drawn down by leaves that have not yet unfurled. A breath exhaled in silence by stems that barely rise above the soil. A breath shared between stone and spore, between mist and moss. The world is quiet, but full. Plants have crept across continents in countless generations, their tissues folded against drought, their pores open to morning light, their lignified stalks lifting reproductive structures just high enough to cast spores farther on the wind. And now something stirs in the lineage, not a transformation, not yet, but a deepening of form. The simple axes of earlier plants, once equal in every fork, begin to specialize. A central stalk holds steady, while side branches angle outward. Cells begin to flatten, to broaden, to divide with asymmetry. The first leaves approach, though they are not yet true leaves. The land holds its breath at the threshold of complexity. This is the stillness before ferns. In this interval, morphology experiments with pattern. Some plants develop innations, small flaps of tissue along the stem, non-vascular but photosynthetically active. Others evolve microfills leaves with a single vascular strand, narrow and efficient, born not from flattened branches, but from outgrowths reinforced by primitive conducting tissue. These are not broad leaves, not yet the complex organs of flowering plants, but they offer surface area light capture, a new ecological presence. With them, the plant body becomes more expressive. Light no longer strikes stems alone. It spreads across angled planes. Shadow deepens. Photosynthesis increases. Spores multiply. Height follows. And with height, the demand for support and transport intensifies. Lignin thickens. Roots grow deeper. Vascular bundles arrange with greater sophistication. The plant becomes a system, no longer a series of cells aligned in function, but a body of differentiated parts, each performing a task, each woven into the whole. The first true ferns remain close. They are not yet abundant, not yet dominant, Their fronds are coiled in tight spirals, fiddleheads waiting to unroll in humid air. They will come soon, will spread across floodplains, and river deltas will rise among horsetails and club mosses and seedless trees. But for now, their presence is embryonic, barely a trace in the fossil record. Their ancestors, the progymnosperms and trimerophytes branch with increasing complexity, form spores in specialized clusters, begin to resemble the diversity that will soon sweep the land. Evolution hesitates at this juncture not from uncertainty, but from the sheer inertia of what has come before. 1 billion years of aquatic life has led to this. Every adaptation built on another. Every trait inherited or altered. There is no clean break between moss and fern, between charophyte and lycophyte. Only lineage, only continuity, only the slow layering of difference. Geologically, the continents stretch wide and the greening continues. In equatorial belts, vascular plants spread through wetlands, forming dense tangles along riverbanks and deltas. In uplands, early roots break down granite and gneiss, releasing minerals into the water table, contributing to the weathering that feeds chemical cycles already altered by plant respiration. Soil profiles deepen, organic horizons thicken, decomposers diversify, and with them come the first detritivores on land, arthropods that crawl through leaf litter, millipedes and mites, and ancestors of springtails feeding on decay that did not exist before the colonization of the land. The food web begins to stratify. Life is no longer just plant and microbe, but consumer and decomposer cycle and return. Yet for all its activity, the moment remains suspended. The threshold is not yet crossed. Seeds have not evolved. Flowers do not exist. Vertebrates have not yet walked the land. The forests remain short, though some trees approach heights of 10 meters. Most of the land remains dominated by low, resilient mats of vegetation, by lycophytes and zosterophils, by mosses and hornworts. In the corners of this ecosystem, in the folds of floodplains and the damp niches of sheltered outcrops, something gathers. Complexity thickens. Genetic potential accumulates. And in this gathering, the planet prepares for what comes next. There is irony here, soft but resonant, that the most profound botanical revolutions begin not with explosions of form, but with the smallest shifts in symmetry in vein pattern, in the arrangement of cells around a sporangium. The appearance of a new leaf type, the refinement of root branching, the bifurcation of vascular tissue. These are the molecular preludes to the forest. From a distance, the land looks still. But within the genome, within the growing tip of each stem, change pulses. Scientific traces of this moment are sparse. Fossils from the late Devonian preserve stems, root spores, and the impressions of early leaves. Whole plant reconstructions are rare, often incomplete. Yet in the gaps, researchers discern patterns. Anatomical sections reveal secondary growth, the thickening of stems through cambium activity, a trait once thought exclusive to trees, now seen in smaller plants preparing for height. The fossil Rhacophyton shows complex branching. Archaeopteris forms wood, but still produces spores. The line between fern and seed plant begins to blur. The lineage forks slowly and without witness. Modern ferns carry the legacy of this moment in every spiral of their young fronds, in every divided leaflet, in every spore capsule that opens with tension and hurls its dust into the air. Their ancestry stretches back through uncounted generations of plants that never bore a seed, that never closed their reproductive cycles from wind and chance. And yet they thrive. They persist in the understory of rainforests, on cliffs in wetlands, in the cracks of buildings and the corners of forests, reminding us that the future was never the only path forward, that ancient strategies refined and resilient still hold. And so the Devonian ends, not with a bang, but with a quiet proliferation. Complexity is seeded across the land in root and frond, in the architecture of stems and the layering of tissue. The earth prepares for forests, for seeds, for insects that fly and vertebrates that walk. But for now it waits. A stillness holds the air. The land is green, not with grandeur, but with potential. And in that potential life leans forward not in haste, but in readiness. Now the light softens. The story of land's first greening long and slow as the breath of stone settles into stillness. No final act, no flourish, just the quiet hum of presence left behind in the pores of moss, the spiral of fern, the dust of ancient spores that once drifted into the silence of air. The continents remain, but they are no longer bare. Rock has taken on memory, soil identity, air pattern. The invisible threads that once tethered life to water have loosened, reformed, rooted in place. Nothing here moves quickly. The winds are slow. The plants do not speak. And yet in their stillness, a world is made. One cell thick, one step inland, one stubborn stalk, lifting upward, each enough to begin. Time stretches not as a passage, but as a presence. Millions of years as a moment held between tide and leaf. What was algae is now land. What was water is now story. What was barren is now quietly alive. The earth does not notice, but it changes. And beneath our feet, in the deep silence of stone, turned green that change continues.